This method can help answer key questions in the fields of macrophage and dendritic cell biology about the subcellular localization and dynamics of various proteins in these cell types. The main advantages of this technique are that it is relatively simple, inexpensive, and demonstrates a better efficiency and stability of expression than the majority of other available methods. Demonstrating the procedure will be Jarmila Kralova, a grad student from my laboratory.
To produce the retrovirus, plate a single-cell suspension of platinum eco-packaging cells in a 10 cm Petri dish and cultivate the cells in 15 milliliters of DMEM supplemented with 10% fetal bovine serum, or FBS, until the culture is 50 to 60% confluent. The next day, add 20 micrograms of the retroviral construct of interest and 10 micrograms of PCL eco-packaging vector to 1 milliliter of DMEM without serum with gentle mixing. Add 75 microliters of PEI in 1 milliliter of DMEM without serum and antibiotics to a second tube for a 5-minute incubation at room temperature, and mix the contents of both tubes together for an additional 10-minute incubation at room temperature. Next, carefully replace the medium on the platinum eco cells with 8 milliliters of fresh 37°C DMEM supplemented with 2% FBS, and carefully add the transfection mixture in drops to the plate. After a 4-hour incubation at 37°C, replace the supernatant with 10 milliliters of 37°C DMEM supplemented with 10%.